Well, good day to everyone joining us and welcome to today's X Talks webinar. Today's talk is entitled Enhanced Peptide Manufacturing Using Backbone and Protecting Groups. My name is Samya Shashikumar and I'll be your X Talks host for today. Today's webinar will run for approximately 60 minutes and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel and that can be found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I would also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who registered on xtalks.com. At this point, I would like to thank PolyPeptide Group who developed the content for this presentation. PolyPeptide Group is a specialized contract development and manufacturing organization, or CDMO, for peptide and oligonucleotide-based active pharmaceutical ingredients. By supporting its customers mainly in pharma and biotech, it contributes to the health of millions of patients across the world. PolyPeptide serves a fast-growing market, offering products and services from pre-clinical through to commercial stages. Its broad portfolio reflects opportunities in drug therapies across many therapeutic areas and with a large exposure to metabolic diseases, including GLP-1. And now I would like to introduce our speakers for the event. Dr. John Karras has 25 years of experience in peptide chemistry. Prior to completing his PhD at the University of Melbourne in 2016, he worked at several peptide manufacturing organizations and startups. He commenced his independent research career in 2022 through a fellowship from the Australian Research Council. His current work focuses on peptide synthesis methodology, insulin analog design, antimicrobial peptides, and enzyme stabilization. And Dr. Linda Haugaard-Kedström has over 15 years of experience in peptide chemistry. She earned her PhD in organic chemistry from Linnaeus University in Sweden and later held a research fellowship at the University of Queensland in Australia. After several years at the University of Copenhagen, where she became an associate professor, Linda transitioned to the life sciences industry. She now serves as Director of QC and Development at PolyPeptide Sweden, focusing on process development for peptide API manufacturing. And Sam Paravizzini, who completed his undergraduate studies in 2017 at the University of Melbourne, majoring in chemistry. He earned his master's degree in 2021 under Professor Uta Wille, studying new urease inhibitors to improve fertilizer efficiency in Australian agriculture. He is now pursuing his PhD under Dr. John Karras, focusing on developing acyl-labile amide backbone protecting groups to improve the efficiency of FMOC solid phase peptide synthesis. And now, without further ado, I would like to hand things over to our first speaker. John, you may begin when ready.
Thanks so much, Sumya, for that very kind introduction. So, lost my screen, sorry.
Oh, we see your presentation structure or slide, John.
Okay,
okay, yeah.
great, thank you. Sorry about that. So today the key objectives of this webinar are to first provide an overview of the state of the art of backbone protection in SPPS, and we're also going to introduce various backbone protecting group strategies. We'll identify capability gaps and offer some potential solutions to our problems for peptide synthesis. We'll also discuss backbone protection in the context of large-scale peptide API manufacturing. And finally, we will give a brief summary and talk about some future directions. So before we get into the webinar though, we thought we'd run a couple of quick polls just to determine who's tuning in today. So this will help us determine whether they're from industry or academia and what sort of peptide chemistry you're working on, such as large-scale peptide synthesis, lab-scale peptide synthesis, or peptide libraries, or even native chemical ligation. So Soumya, are you able to activate those polls, please?
Thank you so much, John. That's right, audience members. We have 2 poll questions for you to interact with in real time. Now, if you are attending via GoToWebinar, the poll is appearing on your screen now, and you can vote by clicking on any of the answers. The question we have for you is, in which setting do you work with peptides? And your answer options are, as an industry, is it R&D, manufacturing, QA/QC, academic research, or other? Again, that question is, in which setting do you work with peptides? Thank you so much, audience members, for voting. I can see that most of you have voted, so I'm going to go ahead and close the poll and share the results. And a whopping 71% voted for R&D, 18% voted for manufacturing. We have none for QA/QC, and we have a 6% tie for academic research and other. Thank you so much, audience members, for voting. I'm going to go ahead and share another poll here. We'll hear from our speakers in a moment. The second question we have for you is, which is your focus area regarding peptide synthesis? Your answer options are lab scale peptide synthesis, less than 50 amino acids, peptide library synthesis, chemical protein synthesis, more than 50 amino acids, large-scale peptide API synthesis, or not applicable. Again, that question is, which is your focus area regarding peptide synthesis? Your answer options are lab-scale peptide synthesis, less than 50 amino acids, peptide library synthesis, chemical protein synthesis, more than 50 amino acids, large-scale peptide API synthesis, or not applicable. I'm going to give a few seconds for the audience members to vote. Amazing, it looks like most of you have voted. So I'm going to go ahead and share these results as well. And it looks like 50% voted for large-scale peptide API synthesis, 17% voted for chemical protein synthesis, 17% also voted for not applicable, 11% voted for lab-scale peptide synthesis, and the final 6% voted for peptide library synthesis. Audience members, thank you so much for voting. John, it's back to you.
Thank you, Soumya. So I've lost my screen. There we go. Yeah, so that was a nice result. So we've got a broad scope of audience members, so many from industry and a few working in native chemical ligation and lab scale synthesis. So hopefully you'll all get something out of today's webinar. So, sorry, I keep losing my screen. There we go. So, of course, FMOC SPPS is the gold standard method for the routine synthesis of peptides. Sorry, I just keep, there we go. Essentially it involves the sequential assembly of amino acids on a solid support, and this involves this iterative coupling and deprotection cycle, and then after you assemble your peptide, you can cleave it off the resin with acid. So this has enabled us to prepare a range of novel peptides, such as macrocyclic peptides, non-native substitutions, and this includes being able to prepare peptidomimetics to improve the drug-like properties of peptides. We can also introduce D-amino acids to prepare mirror image peptides and proteins, which is really important for racemic protein crystallography and also mirror image phage display technologies. We can also introduce site-specific modifications such as phosphorylations and sulfations. We can also introduce fluorescent probes for microscopy and isotopic labels for NMR experiments and quantitative MSMS. We also, of course, need to prepare the peptides for native chemical ligation, and also we can use now with these advanced peptide synthesizers, we can assemble proteins via a single shot synthesis, although you do need specialized equipment to do that. Of course, peptides are also used as biomaterials, such as hydrogels and nanoparticles. And of course, we also use FMOC-SPPS to prepare our peptides on a very large scale, because this is how we prepare our drugs. So as you can see, the projected market's going to increase substantially, and this is going to increase the demand for longer peptides, more complex proteins, and of course we want to be able to make a larger number of analogues to expedite drug discovery. So of course FMOC-SPPS has served us very well over the years. Of course, Bruce Merrifield, he obtained a Nobel Prize for solid phase synthesis, but it still does have its challenges, and the main fundamental challenges are, of course, aggregation and chain solubility. And this occurs during solid phase assembly and also in solution. And this, the aggregation occurs via beta sheet formation or other aggregates where you form these hydrogen bonds. There's also, of course, side reactions that still bedevil us, particularly aspartamide formation, and especially now that we're using heat-assisted SPPS, this, as well as accelerating our coupling and deprotections, we also accelerate our side reactions. And this limits the routine access to peptides more than 40 amino acids in length. Of course, we can make longer peptides, but it's very difficult to do it routinely. We also need to consider the fact that we need to replace DMF, and that's already underway in Europe. So we need to consider whether the new green solvents will solvate the peptide chain as effectively as DMF does. There are some current solutions, of course, and I alluded to the new peptide synthesizers earlier. So, of course, we have microwave-assisted SPPS, infrared heating is also another option, and more recently, the continuous flow synthesizer, which has enabled us to assemble longer peptides and small proteins via a single shot synthesis. Although, as you go longer, the synthetic failure rate also increases, of course, so it's not a perfect solution. We can also introduce polycationic solubilizing tags, particularly at the C terminus of the sequence, with the linker, of course, that's cleavable, and this also improves the solubility of the peptide chain. We can use chaotropic salts such as lithium chloride in the DMF to suppress the aggregation, and of course we can add additives to suppress aspartamide. But we still need to address the fundamental problems that relate to solid phase peptide synthesis and solution phase handling, and that is the aggregation and solubility issues. To address these issues, backbone protection groups have been used, so over the last 40 or so years. So these have several benefits. So first, you can increase your chain solubility in organic solvents. You also suppress aggregation, especially beta sheets. You can actually directly block the hydrogen bonding that's essential for stabilizing the beta sheets. It also blocks aspartamide for asp-glyce sequences, and you can promote macrocyclization because a tertiary amide, which is the backbone, how the backbone protection works, this actually disrupts the inherent linearity of the peptide chain, and it brings the reactive groups closer together, and therefore you get a much more efficient cyclization. We can also conduct epimerization-free fragment condensations by protecting the C terminus with a backbone protecting group. We can also modify the C terminus using a backbone amide linker strategy, and of course we can improve solution phase handling, and we'll talk more about that shortly. So the preferred features of backbone protecting groups are as follows. So first, they must be cost effective to synthesize, so they're available to everyone at an affordable level. They obviously should be FMOC-SPPS compatible. FMOC-SPPS seems to be the dominant method now. We also need to be able to incorporate them really efficiently and also cleave them very efficiently. So they should be really acid labile. ideally they should be able to be universally applied, so we can introduce them in more and more dipeptide junctions. And of course, we want to be able to automate the process so we don't have to do all these manual steps that can slow down our workflows. So in the mid-1980s, Nakano and Ishikawa published this paper on these polyleucine peptides. So basically these peptides formed beta sheets, so they were forming intermolecular beta sheets. But when they were introduced in a tertiary amide, so they were using proline, which is a natural tertiary amide, and also the dimethoxybenzyl group, they found that this beta sheet structure was disrupted. Moreover, they found that the peptides with tertiary amides were much easier to solvate. So they figured that this was a good way to improve the solubility of peptides in general and block the aggregation and apply it to more synthesis. So DMB can be introduced via reductive amination with the corresponding benzaldehyde, and of course you can also introduce it with FMOC monomers and also FMOC dipeptides. This electron-rich ring is TFA labile with the appropriate scavenger, so you can use thioanisole, and there's of course others that you can also use. So here's one example of how the DMB group is used. So this is nucleonin. So it's a C-terminal region. You can see here they used, I think, 14 DMB groups. And so this was synthesized about 20 years ago before the real advances in synthesizers occurred. So I think this is a great example of how effective backbone protection can be. I should note though that the DMB lability is can be slow. So for example, for this peptide, they needed cleavage time of 12 hours as opposed to the approximately 2 hours that we normally do. And also with benzyl-based groups, when you do the TFA cleavage, the reactive benzylic cation can alkylate sensitive nucleophilic side chains such as the cysteine side chain or even the thionine and tryptophan. So it's probably not that good to use a lot of DMB groups when you have these sensitive amino acids in your sequence. DMB is also pretty much limited to glycine because of its steric bulk. However, to get around this, Bob Shepard's group in the mid-90s developed the HMB group. So this can be introduced through reductive amination again, as per this reaction scheme on the left here. And then basically, because it's got a 2-hydroxyl group or an ortho-hydroxyl group on the benzylic group, you can introduce the HMB at more sterically demanding junctions through this O-acyl capture. So first you capture the incoming activated amino acid, and then after that's captured, you get this rapid O-to-N acyl shift to obtain your desired amide product. Then you can continue on with your SPPS. So in this example, they made ACP 65 to 74, which is a peptide that's commonly used as a model system to evaluate FMOC SPPS. So in this system, the N-terminal valine, the coupling is typically inefficient, but when you introduce HMB, it is much more efficient. HMB can also be introduced in other ASP-containing junctions to suppress aspartamide formation in, for example, ASP-ASN or ASP-ASP junctions. And of course, the aspartamide problem is more of a problem because we now use heat-assisted SPPS. So it's important to block this side reaction. The trimethoxybenzyl group was also developed. Now this is even more acid labile, but it is sterically hindered, and it's very difficult to introduce it into the peptide backbone, and it's mostly limited to glycine. But another trialkoxybenzyl group is as follows. So you can use it as a backbone amide linker and append your C-terminal amino acid via the N-alpha amine and instead of the carboxylate, and that way you can orthogonally protect the C-terminus of the peptide, then do your FMOC SPPS, and then potentially do an on-resin macrocyclization as per this synthesis here. So this was a really nice study. Moreover, with a backbone amide linker strategy, you can also introduce modified C-termini. So you can introduce aldehydes, esters, hydroxides, and other unusual functionality there. It's also really good for the large scale because sometimes when you use a Wang linker, you get this 4-hydroxybenzyl artifact. So that's part of the Wang linker that hasn't been fully cleaved from the peptide. So instead of using the Wang linker, you can use this strategy here, and you still obtain a C-terminal acid. Although for this strategy, you must be very careful because you can form diketopiperazine after you deprotect the second amino acid. Pseudoprolines, they're probably the gold standard regarding backbone protection. So these can be synthesized in very high yield, numerous ways. They're commercially available, and of course, now the cost has come down substantially. So these are introduced into serine and threonine containing dipeptides. You can also introduce the acetonide group into cysteine, but now 2,4-dimethoxyphenyl is preferred because it's more acid labile. So here's a nice example of a long peptide that was synthesized about 20 years ago again. So this is a single shot synthesis of this fast death domain fragment. They're using 8 pseudoproline building blocks, so you can see how effective that is. So this would not be accessible without the use of backbone protection. Pseudoprolines are also really useful to promote macrocyclizations because, as I said, the tertiary amide promotes more of a cis conformation about the amide bond, and therefore both termini are more proximal, or the reactive partners are more proximal. So this is a really good example here. This is a sarafatoxin analog, and you can see here it's assembled using 4 pseudoproline building blocks. And then after the TFA cleavage, you retain this oxazolidine because this one is actually TFA stable. But it also promotes formation of the disulfide isomer with the correct connectivity. So of course, in this example, you could potentially get 3 isomers, but because of this turn-inducing moiety here, the pseudoproline, then you obtain the correct isomer. Similarly, the Robinson group used a pseudoproline to promote a ring-closing metathesis reaction on the solid support, so using Grubbs catalyst, and then after that, you can simply do your TFA cleavage and obtain your macrocycle in excellent yield. Pseudoprolines are also useful for fragment coupling. So this is work I did quite a few years ago now. So we were making teixobactam. So this is an antimicrobial peptide. It contains an N-terminal chain and a C-terminal macrocycle. So what we did, we made these 2 fragments separately, and then we ligated them together. And of course, with the N-terminal fragment, we had a pseudoproline here, and this suppressed epimerization because it suppresses formation of the oxazolone intermediate that leads to epimerization. So although this coupling was really difficult, we didn't see any epimerization. This is another interesting study where you can obtain pseudoproline-like moieties through this ligation via a C-terminal glycol aldehyde. So you need either a serine threonine or a cysteine at the N-terminus of this second peptide, and you have your aldehyde here. So essentially, you can first form the imine, and then you form this oxazolidine, or if it's cysteine, it would be a thiazolidine. Then you get an O to N acyl shift, and you obtain this proline-like moiety here. Of course, it's got this hydroxymethyl group, but it's still a very good proline mimetic. So there's also other serine and threonine-based dipeptides with backbone protection. So this is work done by Suechen Li's group very recently, in fact. So first you can take your FMOC amino acid and you form this salicylaldehyde ester. then you can attach that to a second amino acid and form this N-benzylidene. And these can be obtained in really good yield. The allyl group can be deprotected in situ just prior to the coupling. So you can obtain these in 2 steps. They use these building blocks to synthesize a range of proteins, including this synthesis of ubiquitin via a single shot synthesis. So they used 3 N-benzylidene moieties here, and they were able to obtain this in really good yield. And one feature of these N-benzylidines is that they seem to perform superior to pseudoprolines, which is probably due to this pronounced kink that it induces in the peptide backbone. So other serine and threonine-based dipeptides include the isoacyl dipeptides, which was developed by Kiso in the early 2000s. So essentially, we are isolating the side chain of a serine or threonine residue to form this deoxy dipeptide. These are commercially available, and their price again has come down substantially. And they're really useful because you can retain this deoxy bond even after cleavage, and this disrupts the aggregation propensity of peptides because you have this free rotation here, and it disrupts the inherent linearity of the backbone. And here in this example, this is amyloid beta 42, so you can introduce also this deoxy bond in situ. And then, of course, you have a Boc group protecting the N-alpha amino group here. So after you introduce the deoxy bond, you can do more SPPS and then do your TFA cleavage. So as I said, you retain the deoxy bond, and then you can do your purification and then get a really high purity, and then you can simply do an O to N acyl shift to generate your native peptide backbone. You do have to be careful here though, because sometimes you can get a beta elimination during assembly, which occurs during the FMOC deprotection steps. You can also conduct epimerization-free fragment coupling. So this is done on the solid support, but of course you can also do it in solution. So you can introduce this peptide without epimerization because you've got urethane type protection here instead of this N-acyl. This being N-acylated, so this suppresses the oxazolone formation again. And then you can do your TFA cleavage, and then finally do your O to N acyl shifts to obtain your native backbone. So those isoacyl dipeptides are really useful, and we use these routinely to prepare insulin analogues because they also have a high propensity for forming fibrils. So people have developed other heterocyclic backbone protecting groups. So Bob Shepherd also looked at furfuryl and 2-thionyl methyl groups. So these were found to be really acid labile, but they were difficult to incorporate and also isolate. However, because of their high acid lability, I feel like there's scope for improvement there, and this could be a new avenue of discovery. Similarly, the Albericio group developed this E.N group and also this indole-based group here, and they were also found to be quite acid labile compared to DMB, and they were able to suppress aspartamide, and they did find that, however, that the E.N was better than this N-methylated indole-based protecting group because this is a little bit more sterically bulky. The dicyclopropyl methyl group is also really acid labile, and it can be used at gly and allo junctions. And I feel this is a really good protecting group, I think, but maybe it's been a little bit underutilized. So these can be accessed from this ketone, and then you can convert that to the dicyclopropyl methanamine. You then do a transimidation, and then you can do a reduction to obtain your secondary amine. After that, you can cleave your ester and then finally protect the amino group with FMOC or even Bismoc. So Bismoc is like FMOC, but it's even more base labile. So they use the dicyclopropyl methyl group to make the PRP 106 to 106 peptide, which is another model system that we commonly use, and they were able to obtain this in very high yields, crude yields, so it was very impressive, really. They were also able to suppress aspartamide again, which is very important, and they also introduced it into long polyalanine sequences, which of course are very polar and they really drop out of solution and aggregate. So this was really nice work by Carpino and Binet. So I'll hand you over to Sam, who's going to now talk to you about the tetrahydropyrenyl group. Thanks, Sam.
Thanks for that, John. So today I'll be talking to you about the THP group of tetrahydropyrenyl. This has been commonly used in organic synthesis as a hydroxyl, amine, or thiol protecting group. But for us, we decided to use it as an amide backbone protecting group to help improve the solubility of peptides in SPPS. We were able to install the THP group into numerous dipeptide junctions through an acid-catalyzed activation of 3,4-dihydropyran, which installed the THP group onto an amino acid through addition. This protected amino acid could then be acylated using a mixed anhydride activation method with isobutyl chloroformate, producing the protected dipeptides in up to 80% yield. The benzyl esters were then removed using hydrogenolysis with palladium hydroxide, and this was able to produce our THP protected dipeptide building blocks. We also made sure that the THP group was going to be compatible with FMOL cleavage conditions, and so was subjected to 20% piperidine treatment, in which we found no significant degradation. We also made sure that there was no detectable amounts of epimerization at either of the alpha carbons on the building blocks, ensuring that they maintain good stereochemical integrity during their synthesis and during their application in solid phase. So next, we needed to make sure that the THP group was highly acid labile, and we wanted to compare it with other known groups like the DMB and pseudoproline groups. And we were pleased to find that in 10% TFA, there was a clear trend in where the THP group was significantly more acid labile than the DMB group. We also found that in 1% TFA, the THP group was stable, which is beneficial for the synthesis of protected peptide fragments and for the fragment condensations, improving the solubility of the peptide in solution. We also compare the THP group to the dicyclopropylmethyl group, and we were pleased to find that on this model structure here that it was easily removed in as low as 2% TFA. So with our dipeptides in hand, we went on to synthesize a couple of different sequences, namely the prion peptide, as mentioned before. When we attempted to synthesize this without backbone protection, we only got the target in very low purity, around 4%, and this was increased to around 19% when we used one THP group on the backbone. We were also pleased to see that this was significantly increased to 33% purity when using 2 THP groups to help improve the solubility of our peptide. We also synthesized nigrosin, this alpha helical antimicrobial peptide called nigrosin HLM, and we were pleased to see that with THP backbone protection, we could improve the crude product purity to over 50%. This translated to almost double the isolated yield after purification of the sequence. The THP dipeptides were bench stable crystalline solids, and we were able to produce them in decent yields. We proposed that because of THP's high acid lability, it is a potential replacement for DMB specifically at glycine containing junctions. So thanks, I'll hand it back over to John now.
Thanks, Sam. Now we'll move on to sterically small backbone protecting groups. So these are interesting because of their low steric bulk, so they could potentially be introduced at more hindered junctions. So the Albericio group evaluated ethoxymethyl groups, so they were able to produce this alloc protected dipeptide and introduce it to the solid support. And followed by that, they cleaved the alloc group and found that they also cleaved this ethoxymethyl group. However, this other ethoxymethyl group was retained, and then they were able to continue their synthesis and improve the assembly. These are very difficult to incorporate and synthesize However, because of their high acid liability and of course their small steric bulk, I feel like they've got a lot of potential if we can improve their synthesis. Other sterically small backbone protecting groups include the propagyl group. So this is retained post-TFA cleavage, and it can therefore improve solution phase handling. So it's introduced via the submonomer method, which is analogous to how peptides are produced. So essentially you can isolate the N-terminus of the peptide on the solid support using bromoacetic acid, followed by alkylation, and then you can isolate the amino group and then do further FMOC SPPS, and then finally you can TFA cleave the peptide and still retain this propagyl group. This can later be removed with gold 1, so that's quite convenient and orthogonal, although you do have to be careful because sometimes you can get cleavage at this tertiary amide prematurely. Other backbone protecting groups that are retained post-TFA cleavage include photocleavable protecting groups. So this is really nice work by the Aylward group. So these are used to promote macrocyclizations. So essentially, in this study, they made this tetrapeptide macrocycle. So they did this by introducing 2 orthonitrobenzyl groups. So of course you need the nitro group in the ortho position compared to the benzylic position, so you are able to photocleave it. So they've introduced one here where they can, which can promote more of a cis amide conformation about the amide bond to bring the 2 reactive termini together. And then they introduce a second one on the N-terminus. And note that as well as the nitro group, they've got a hydroxyl group in the other ortho position. And in this way, they're able to capture the C-terminus via this ester intermediate, and then they can do a ring contraction and obtain the really high energy macrocycle here. So when they tried to do the photocleavage of the protecting groups at the end, they did find though that they were getting an N to O acyl shift back to this intermediate, the previous one. Therefore, they had to methylate the phenolic group, and after that they were able to obtain their 12 atom macrocycle, which is quite an achievement, I thought. And of course, there are other photocleavable protecting groups that you can use for the backbone, particularly formethoxy 2-nitrobenzyl, which has been developed by Kent and co-workers. There's also been backbone protecting groups bearing safety catch groups. So this is a way to tune their acid liability by modifying aryl substituents. So essentially you can reduce the electron density of an aryl group and that will increase its TFA stability, or alternatively you can increase the electron density and then decrease its TFA stability, or i.e. make it more TFA labile. So this can be done several ways. So one way is to introduce a nitro-containing benzyl group into the backbone, and then so this will be retained after the TFA cleavage, and then that gives you the benefit of suppressing aggregation and improving solubility. And then you can, after you do your purification, or if you're doing native chemical ligation, then you can simply reduce the nitro group to an aniline, and then that becomes more electron donating, and therefore it becomes more TFA stable. Similarly, you can do the same thing with a sulfoxide containing benzyl group and convert that to a thioether via a reduction again. And of course, you can acetylate the phenolic group to ensure that it's retained after TFA cleavage, and then hydrolyze it later, and then cleave it off with TFA, because it's simple. It's pretty much a HMB group. So in one example, the Albericio group synthesized amyloid beta 42. So they introduced this sulfoxide containing benzyl group on the backbone. So after they were purified it, the amyloid beta peptide in very high yield and in excellent purity, they then simply reduced the sulfoxide to the thioether under mild conditions, and then they were able to remove that, and they were able to obtain this peptide in very high yield. So anyone who's worked with this peptide would understand how difficult that is. So safety catch backbone protecting groups can also be introduced through native chemical ligation. So in this example, this is work done by Swetch and Lee's groups using his serine threonine ligation technology, and in this case, he's using cysteine here. So essentially, you have this cellulosilaldehyde ester, and you can introduce that. And then after that, you can do your FMOC SPPS. And note here, also appended to this protecting group is this alloc protected amine. So essentially, what he did was complete his FMOC SPPS and then cleave off the alloc group, and then do more SPPS to introduce all these histidines. So essentially, he generated this polyhistidine tag, which is obviously going to be used as a solubilizing group. You can then do the cleavage with TFA, but before you do that, you must acetylate this backbone protecting group here, otherwise it will be TFA labile. So when you do that, of course, you retain it. This acetoxy group, of course, is hydrolyzed during further native chemical ligation. So you don't really need to do a separate step. And then after you've done all your native chemical ligation and other synthetic transformations in solution, you can simply just cleave this off with TFA to obtain your protein of interest. Of course, there's many other similar methods to this, and one in particular using a polyarginine tag was conducted by Lei Liu's group. So these can also, we can also functionalize backbone protecting groups with reactive ligation partners to promote native chemical ligation through proximity effects. So what I mean by that is we can introduce, for example, these two protein fragments which rapidly self-associate. So this is work done by Lei Liu as well. And essentially you have this one peptide fragment which has a thioester, and you have another fragment, of course, with the N-terminal cysteine. But to promote native chemical ligation, even in high dilution, you can first form this joins together to form this larger protein. And after that, you get this really rapid NCL through proximity effects, because both the thioester and the cysteine are close together, and like so. And then, of course, you can again cleave these backbone protecting groups off with, in this case, HCl and HFIP, which is an alternative to TFA. Of course, we'd also like to introduce backbone protecting groups through automation, just to speed up our workflows. We can obviously do that easily with FMOC monomers and dipeptides containing backbone protecting groups. But there's another really nice study done by John Offer's group about 10 years ago. And basically, they modified their peptide synthesizers so they could conduct reductive aminations and do those in situ, in an automated fashion. So essentially, they've got this sulfoxide-based benzaldehyde, so they can introduce that and then through reductive amination. And then after that, note also there's a hydroxyl group also to the benzylic position. So again, they can introduce the next amino acid via this O-acyl capture. And then of course, you can do your O to N-acyl shift. So that means again that this protecting group can be introduced at sterically demanding junctions. Also note that you have this electron withdrawing sulfoxide group here, para to the hydroxyl group. So this makes this a much better leaving group. So you get this rapid O to N-acyl shift. Finally, you can do more SPPS, and you have this option here of either retaining this protecting group after the TFA cleavage, if you keep it as a sulfoxide because it's electron deficient, or you can convert it to a thioether where it's more electron donating, and then it will cleave off with all the other side chain protecting groups. So there, as you can see, there's quite a number of options that we have for backbone protection. However, there are some capability gaps, so it's really important to try and identify them. So remember that, of course, we need to more reliably access longer peptides more consistently, so we can turbocharge analog development and even access longer and longer proteins via NCL, so we can improve the scope of our peptide chemistry. Pseudoprolines are, of course, the gold standard and highly effective, but they're not suitable for aliphatic-rich segments. And of course, that's often where the problem occurs in terms of insolubility and aggregation. DMB is very effective, but of course, you still have these cleavage issues. So you don't really want to use a lot of DMB groups if you have a disulfide-rich peptide, for example. So ultimately, we would like to develop a backbone protecting group that is more acid labile than DMB and more easily scavengeable than DMB. Also, we'd like to introduce dipeptide building blocks with backbone protection, so we have more options to use backbone protection. So as you can see with some of those examples, the more we use, the more effective our synthesis is. So on the right-hand side here, this is a table of the natural abundance of all the dipeptides. Now, I must stress that this is not perfect. This data originates from the UniProt database, so it does assume that each amino acid is randomly distributed throughout the proteome. Of course, this is not totally true, but I think it's a very good guide to help us determine which which dipeptides are the most abundant. So as you can see here in blue, we have all our pseudoprolines, and I've also included proline, which gives us natural backbone protection. In purple, we have glycine. So of course, currently we're using DMB gly, but we want to improve on that because we're not really able to introduce DMB all the time at glycine because of these limiting factors that I talked about earlier because of the cleavage issue. We've also identified 18 dipeptides that are highly abundant. So these 18 in green are at least 0.5% abundant. Also, they don't contain cysteine, proline, serine, threonine, or glycine because we already have solved those problems for those dipeptide junctions containing these amino acids. And also these ones that we've selected, they don't contain beta branch side chains. So that means that they're more feasible to synthesize with backbone protecting groups. So I feel that if we can make these, then we can increase our sequence coverage to 37% based on abundance. So that may be all we really need to solve a lot of our synthetic problems. So what are the backbone protecting groups with high potential for future use? So first, I think the dicyclopropyl methyl group is quite acid labile, and I think it's quite underutilized. So that could be a potential replacement for DMB. Of course, the THP group that we recently developed, that is hyperacid labile compared to all the others, but we still need to optimize the synthesis of these THP protected dipeptides, so that work is not quite there yet. Other N-acetyl groups could be quite good if we can make them effectively, but at the moment they're quite difficult to make, but they are superacid labile and sterically small. And finally, I think there's a lot of scope to investigate heterocyclic backbone protecting groups. So just some tips for the lab scale researchers. So I feel that every lab should have this compound in their cupboard, 2-hydroxy-4-methoxybenzaldehyde. It is super versatile. It can be introduced readily through a reductive amination in situ. It can be introduced at hindered junctions via this O-acyl capture method that I described previously. And also you can retain it after cleavage by acetylating the phenolic group. So I think it's really excellent for the lab scale. It's not so suitable for the large scale because you introduce a competing nucleophile with the N-terminus. So any subsequent couplings, you'll deplete your incoming valuable FMOC amino acid rapidly. And of course, in the large scale, you're using fewer equivalents of amino acid. I think these pseudoproline monomers are also excellent because it means that you don't have to buy all the pseudoproline dipeptides, which can be quite expensive. You can simply just buy the serine and threonine monomers. However, you do have to be super cautious about their incorporation because this unmasked amine is not as nucleophilic as primary amines. And of course, you have the steric hindrance of the acetonide group. And finally, I still think that DMB is really useful if you only need to use one or two. And of course, that's sequence dependent. Of course, ultimately we want to replace it, but it's still very useful if you're going to use it on a limited basis. So I'll now pass you over to Linda, who's going to talk to you about large-scale SPPS.
All right, thank you, John and Sam, for so elegantly going through the available backbone protecting group strategies. So as John mentioned, let's consider large scale. Let's consider industry setting. Let's discuss, instead of lab scale, let's discuss 100 grams, kilograms, 10+ kilograms of peptide per batch. So in general, we could say that there's 4 categories that should be considered. So there will be manufacturing and operational factors, but also supplier and regulatory considerations. But if we go through and start with the manufacturing aspects. So of course, from an industry perspective, choosing the correct backbone protecting strategy needs to be related to cost. So John mentioned these special starting materials, as we say in the industry. So that is like the FMO protected amino acid with some kind of protection on. They are called starting materials. So you will hear me talk about this quite often. But they are normally more expensive compared to the standard amino acids. So this always needs to be considered when you choose to introduce a protecting group or not. But in reality, if you have some challenges, like John mentioned, in terms of aggregation, or it's difficult to solubilize the peptide on the solid phase, then of course this strategy should be considered. And it could also be that you can reduce the impurities formed by this strategy. And not only can you improve the yield by using some of these tools, it could also potentially be that by reducing impurities, you can also make the downstream processes a lot easier. So for example, if you have less impurities, potentially you can remove one purification step or more. It can also mean that if you have less impurities, that you can increase the efficiency of the purification steps by loading more onto your column and thereby reducing the time it takes to manufacture your peptide. Again, cost is important when it comes to large scale and industry setting. And another factor in terms of the manufacturing aspect is that if you have less impurities, you might have to be able to do less fractionation and again reduce the time of the overall manufacturing. In terms of specification and quality control, we know that health authorities across the globe are of course very focused on the quality, and this is controlled through specification, both specifications of the final peptide, the final API, but also specifications in terms of the starting material. And here we need to pay really good attention. We need to develop methods to make sure that we can separate all impurities that are formed during the ribosynthesis for these backbone protecting starting materials. But this is something that is, standard operations in terms of foreign industry. But again, it should not be ignored. It takes a lot of time to get these methods out to verify that you don't introduce any new impurities related or coming from the starting materials into your peptide. And of course, these protecting or the special backbone protecting starting materials need to be able to be handled in bulk, and they need to be stable at various storage conditions so they can be handled in an industry setting. All right, so in terms of supplier and regulatory considerations, so when we talk about very, very large scale batches, of course, all manufacturer needs to have a procurement strategy. So this in practice means that there should be several suppliers for given starting materials. This also includes these special starting materials that we discussed today. And again, here it's important to consider the route of synthesis. So that means how are these backbone protected starting materials actually manufactured? Because when it comes to health authorities, you want to have the route of synthesis from multiple suppliers as equal as you can. And you need to make sure that you don't introduce any impurities arising from the starting material into the peptide itself. So let me take you through one example. So in some cases, these starting materials can be manufactured using acetic acid. So if you're trained as a peptide chemist, you know that acetic acid can actually acetylate your growing peptide chain on the solid support and thereby introducing an impurity. So here you really need to be sure that you have this parameter included in the specifications so you don't end up having large residual acetic acid forming new impurities in your peptide that comes from this starting material. And finally, I would just like to mention the regulatory demand. We know that different health authorities across the globe has different demands in terms of starting materials, and this should absolutely not be ignored. And you need to be very aware of what the different markets, if it's US, if it's EU, or if it's China, or if it's some other market, what demands these health authorities have on these starting materials. All right, so just a short conclusion. I hope we have been able to show you various strategies in terms of background protections. We also discussed the current gap in terms of capability. And also had some suggestion on how we should address this gap in terms of replacing, for example, DMP with more acid label background protecting groups. And we also showed you a couple of examples of new capabilities that could be part of this toolbox going forward. And also discussed what should be considered in terms of new technologies such as heated SPPS and solubilizing tags. And also that when you design your manufacturing for larger proteins, don't forget the enzyme aspects. And we would also like to thank or acknowledge Craig Hutton, also based in Melbourne, that has been part of this work, but could not join us today during this webinar. And with this, I open the session for questions.
Well, thank you very much, Linda, and all our speakers for their very insightful presentation. I know that we are at the end of our webinar, but I think we can take a question or two here.